Hi, good evening. I'm David Levine. I'm co-chair of Science Writers in New York. Um, I want to thank Joe Bonner, who's behind the scenes and who's producing this. And I'll also put it on YouTube for people who can't make it tonight. My guest tonight is Dr. Robert Seder, who became chief of the Clinical Immunology Section in the Laboratory of Clinical Investigation, part of NIAID, Division of Intramural Research of the National Institutes of Health. Dr. Seder was then appointed to a tenured position in the Vaccine Research Center, Laboratory of Immunology in 2000. Since joining the VRC, Dr. Seder has focused his efforts on understanding the innate and adaptive mechanisms by which various vaccines, approaches, mediate protective antibody and T-cell immunity in mouse, non-human primate, and human models of HIV, malaria, tuberculosis infection. And for tonight's discussion, he was involved in the research that led to the approval of Modernis candidate mRNA-1273 vaccine, which was co-developed by scientists at NIAID, as well as Moderna. So welcome, Dr. Seder. I know we had to postpone last week because you're sick, and I know you're still not feeling great. I'm happy you don't have COVID-19, but you have some nasty respiratory virus. So anyway, so why don't we talk? So New York, May 19th is opening up completely. Other states are opening up as well completely. COVID rates are going down in a lot of states. Broadway is going to open in September, New York audience. Even baseball games, all kinds of things, going to have concerts. The CDC guidelines last Thursday said fully vaccinated people, like the two of us, do not have to wear masks outside or even inside if we're with fully vaccinated people. So what do you think of all this supposedly good news and also for some people worrisome news?
Well, so thank you for having me on. It's a great pleasure to be here. So I think that the story of the COVID vaccine is great news. I think it was developed rationally by my colleagues and other people around the world, based on years and years of experience of how you would design a vaccine against an upper respiratory viral infection. And then I think the magic of RNA delivery showed that it can be done quickly. And so I think the data we have so far, as you've seen, is that with Moderna and Pfizer, the efficacy is about 94%. And the Johnson & Johnson vaccine and AstraZeneca is about 70%. But if you extrapolate that to kind of what we consider to be severe disease and hospitalization, all the vaccines, the Johnson & Johnson and AstraZeneca, including the Moderna and Pfizer, are highly effective. So I'm confident in over the next several months that we all have very robust immunity sufficient to mediate anything very bad from happening, such as pneumonia and lower respiratory infection that would lead to hospitalization. And so I think that's all the good news. The question is over time, as the immune response might wane, would we become susceptible to infection? And that could be a mild infection. It could be an upper respiratory infection with a runny nose, or it might be, in the case of like the recent Yankee baseball players who received a vaccine and are getting tested constantly, a positive PCR test, even though they're asymptomatic. And so you can see that I think 7 of 8 Yankees who had received the Johnson & Johnson vaccine felt perfectly well, were getting tested, and came up positive. And so that would indicate that the vaccine was working. They weren't sick, they weren't symptomatic, but they did test positive. And so I think it's important for people to be aware that, quote, these breakthrough cases are really, in those cases, are not associated with clinical symptoms. And so they're perfectly fine and protected. One of the issues is would they be able to transmit that infection? And I don't think we entirely know that yet. Whether, even though they're PCR positive, whether they're able to transmit. But I do feel that all of us who have achieved both vaccinations with Pfizer and Moderna would have a high likelihood of being free to go and live your life and go to movies and go to restaurants and not be in fear of having any serious illness. Now, 6 months from now, it's certainly possible that, as I said before, you still are protected, but you might, in fact, be able to contract an infection that would register positive by PCR, or you might actually have some mild upper respiratory infection. And so, a lot of the studies that we've done and other people have done are now looking at whether you would boost people perhaps over the next 6 months or 9 months, and what that boost would provide you with an additional enhancement of your immune response so that you would have sustained high level protection. And the data that is being generated right now shows that if you wait, people that are originally got vaccinated early in the year and have gotten boosted get a dramatic increase in their immune responses that are actually much higher than the initial vaccine series. So I think it's likely that a boost will be offered, and I think getting boosted will put you well above a protective threshold, and that might actually last for a long time. So all of this is good news. You might need another shot at some point, but I think we're on our way toward normalcy.
So how will you know whether you need another shot?
Well, I can't imagine that we're going to end up doing screening and measuring people's responses. Now, it's possible in the next month, I think there'll be what we call an immune correlate study, which will show that people that got the vaccine, they'll have a metric of antibodies that they'll say is correlated with protection. So if they define that metric, it's possible that you could measure people to see if they've achieved that. And if they haven't achieved that, then maybe you'd say, that would be the time for a boost. I would think that would be perhaps difficult to do scalably. I think the more likely scenario might be that there could be a recommendation in the fall or winter or, another 6 months where they might say that, when the data comes out that boosting really dramatically increases the response, and we're starting to see some new infections, that people would it'd just be a recommendation that we'd recommend that you get boosted rather than testing you prior to the boost. And that might depend on the individual. So as with anything with COVID, it evolves in a minute-to-minute basis where the information is obtained and then released. But I think that's probably going to be a recommendation at some point in the next 6 to 12 months. And that boost might be a little bit different than what you originally received. What we're working on now is maybe you'll get boosted with part of the vaccine you originally received, and then we're going to give you a new variant boost that might match something like the South African variant or potentially the Indian variant. I mean, because of the power of RNA, you can make the RNA match different variants. So you could make an RNA for South Africa, you could make an RNA for India, you could make an RNA for Brazil. The data that's going to come out in the next couple of weeks is going to show data with the combining the current RNA against the Washington strain with RNA against the South African variant. And that data looks very good. So it's possible you'll get what we call a bivalent vaccine. Just like sometimes with influenza, you'll get a quadrivalent vaccine. You might get, 3 or 4 different types of strains. So my sense is that the next boost you'll get might be a bivalent boost to cover you for, what we're doing now is we're monitoring what's circulating globally and determining, whether we need to adjust the vaccines to match the global strains.
So I got the Pfizer vaccine. Will that mean I will have to get the Pfizer vaccine as a boost?
So I think I can't give you a clear answer on that. It's conceivable you could get boosted with another vaccine. You could get boosted eventually if Sanofi or Novavax, which are protein vaccines, get approved. Could you get boosted with a protein vaccine? Possibly. Would you get boosted with a adenovirus vaccine like the Oxford or the Johnson & Johnson? Possibly. I think the greatest likelihood, and what I'm going to do myself, is if you've had an RNA vaccine, I would just get another boost of an RNA vaccine. The data that we've generated, Modernis generated, shows that another boost of the RNA vaccine after a primary series looks fantastic. I'm not aware of data using other platforms following RNA vaccination. Maybe they're even better, but that data has not been generated yet. So it's possible you could have a different one, but I think I would stay with a winner.
Okay. So people who got Pfizer could get a Moderna booster. And
Yeah. In terms of that, I think if you had a Pfizer and a Moderna, I think it's, I would think that would be perfectly fine. Those vaccines are very similar. And so the only issue is whether they're different in the terms of whether they are including a variant RNA, another variant RNA or not. But I probably would feel comfortable that if you were in a place and they were offering a Moderna or Pfizer, I'm not sure that would make any difference at all.
Okay, so for everyone listening in, if you have a question for Dr. Seder, I'm sure everyone does, please put it in the Q&A box. Do not put it in the chat box, and we'll get to questions. Tell us about you were involved with the Moderna vaccine.
Yeah, so the way this happened is my colleague Barney Graham, who I believe there's certain people in this pandemic that are the true heroes of it and the true leaders of it. And Barney is one of them, if not the most important person. So he had been studying SARS and MERS, the Middle Eastern Respiratory Syndrome and SARS before this, and had been developing vaccines against those. And he had also been involved by studying those. He was able to design the spike insert for those infections that he knew would be best in a vaccine. And so when December came along and he saw that there was an outbreak, all he really was waiting for was the sequence of the SARS-CoV-2, where he could take that sequence and then adapt what he had spent years working on with colleagues like Jason McClellan and others to make the spike insert just to match SARS-CoV-2. So on January 10th, he got the sequence of SARS-CoV-2 from Wuhan, and him and Jason designed the insert based on their previous work, where they stabilized the spike with something called the 2-proline stabilization change in the protein. And he sent Moderna the sequence of his design on January 13th, 3 days later. By March 26th, there was a phase 1 trial with the Moderna vaccine. So 2 months later, they already had a phase 1 trial, and by July, there was a phase 3 trial. So, and the reason why is that RNA vaccines are much simpler and faster produced than conventional protein vaccines or adenovirus vaccines. So that's how it really all began. It began with a lot of prior research and kind of being ready to go. And so that was the key step. And all of the vaccines except the Oxford vaccine use his stabilized what they called S2P insert. The Oxford vaccine doesn't. So J&J, Sanofi, both Moderna, both Pfizer, and Novavax all use that.
These were all government. All these people you mentioned work for the government.
So Barney and I have worked 20 years at the government. Yeah. And so he had a collaboration with Moderna because they had been making RNA vaccines. He's interested in flu and RSV and other respiratory viruses. But yes, that was all done within the US government at the NIH.
So I have friends who say, fellow journalists say, well, right now the vaccines are free, but they're going to be promoting boosters in order to make more money.
Yeah, I mean, we don't, I think for those of us who do this every day, the money never, enters into the equation. The government funded all of this. I think that the vaccines, I think they charge, I don't know, $30, I think a vaccine, something like that, $20 or $30. They're given away free. I don't know if the booster would cost anything, but I think if I told you for $20 or $30, you were going to get a boost that was going to increase your immune response tenfold, that would mitigate your risk of getting infected with COVID, I think that that's a very favorable risk reward ratio. I mean, think about it, that,
I think it's great, but I got the shingles vaccine. That did cost some money.
well, the shingles, yeah. So that's kind of, something that we're interested, those of us who do this, we're interested in the solutions, and we want to make the solutions. And then, I think, the companies then roll it out, and that's a separate discussion. I think SARS-CoV-2 has been handled well because the government has basically paid for all the vaccines. But obviously, the companies have sold a lot of product. I will tell you, though, that there is a benefit. So when I, working, with Moderna or the other companies, they take that technology now, and they're going to start to apply it for tuberculosis and malaria and influenza. And so I think there could be a tremendous benefit for other infectious diseases by this technology.
Okay.
Okay.
So I have a question from one of our members says, can you comment on even earlier foundational research that helped pave the way, including DOE funded work at Brookhaven Lab that deciphered the 17 virus RNA polymerase, the key enzymes that enables everyone to make any specific mRNA at scale?
Yeah, so I'm afraid I can't comment on that. It sounds fascinating, and I'm sure there's a lot of government foundational work that went on in other areas that might have contributed to that, but I'm not aware of that specifically.
So please continue to put your questions. In the meantime, I have some questions that were submitted in advance. About how soon after a successful vaccination does the injected mRNA break down? For instance, less than 4 hours, question mark?
I don't know the answer to that. I would bet probably less than, I think that's probably a reasonable guess. How quickly it's gone. Yeah, I mean, that's probably within a 4 to 12 hour time frame.
Okay.
Remember, it's encapsulated in a fat droplet, so it gets into the cell. It's got to express, but yeah, it could be fairly quickly. Could be fair on that order.
Yeah, I've been kind of fascinated by when the vaccines first came out. People said, "I don't want a vaccine based on new technology.
Yeah,
I want to go with a tried and true." Now everybody wants vaccines. They say Pfizer is now the chic vaccine in the Hamptons and stuff.
Yeah. I go by the data. So the data, I think, with Moderna and Pfizer is pretty irrefutable in terms of their efficacy. What you don't have is you don't actually have long-term data. So this is the first RNA vaccine that's been scalably produced. The Novavax and the Sanofi vaccine are protein vaccines. So those are vaccines, as you know, Your shingles vaccine was a protein vaccine. HPV is a protein vaccine. Hepatitis B is a protein vaccine. So those are vaccines for other viral infections that, have long-standing data. But like I said, I took the Moderna vaccine, and both my kids took the Moderna vaccine. So I think that again, based on the technology, the results are quite striking. But over time, we'll generate a much larger data set for whether there's any potential issues over the long term. But right now, it's been very safe in all populations studied, including pregnant women, and including, so far, adolescents, and now they're going down to children.
So someone said, I received sad news from a friend in India regarding deaths of people who received both shots of Pfizer, or at least one in India. One of them, 80 years old, received both vaccines. Another 59-year-old lady received only one and didn't make it to the second. So I would like to know more about the new variants, and when will the FDA authorize booster shots, which we've kind of addressed.
Yeah, so that's a good question. So I'm spending now 24 hours of my day focused on the Indian variants. So the way we look at this is there's a group that's been put together that looks at variants every second of every day. And then they put a priority list of what they're most concerned about. And that's based on whether the variants are spreading, are they transmitting, and then do the variants represent difficulty in the ability of vaccines to neutralize them. And so the reason why we became very focused on the South African variant is that there is much less neutralization against the South African variant than there is against the UK variant or the United States variant. And so we've just recently published, or we're going to submit some data showing that the current RNA vaccines, even those there's a reduction in the immune response against the South African variant, it would still be protective. And so what you can do to remedy that is you can make a new RNA against the South African or the Indian variant. The Indian variant, there's two. It's called B1617.1 and B1617.2. It looks like the B1617.2 is spreading faster, but we don't have a lot of data right now to see, is it neutralization? Is there much less neutralization against it with our current vaccines? We have some data against B1617.1 that suggests there is a decrease in the ability to neutralize that virus. So we'll have more answers probably in the next 2 to 3 weeks. My sense is the current vaccines, given 2 doses, would be effective against those variants. But we were on the phone with Moderna today, and they made a prototype for the B1617.1, and we discussed whether they also make a prototype for the B1617.2. And so they're able to make those quickly, and we can address whether that's needed to incorporate into a vaccine for India. The difficulty is your questioner might realize is that there really are very few RNA vaccines in India. It was mostly the Oxford vaccine, the AstraZeneca vaccine, and then one that was manufactured within India. And so if we can get the RNA vaccines in India, and part of the difficulty was that the RNA vaccines needed to be frozen. So that was a difficult cold chain. But if we could get the RNA vaccines into India, I think that would be an enormous step in the right direction. Otherwise, they're going to have to reformulate, maybe potentially reformulate some of their own vaccines. I also should urge you all that we believe that for the variants, like the South African variant and the Indian variant, you really are optimally going to need both of those immunizations. I know that there's a lot of discussion about whether you just go ahead, give everybody a single immunization, and at some point come back and give them a second immunization. But we have data to kind of clearly show that both vaccinations are really going to be important against the variants.
Okay. When are we going to have FDA approval of these drugs?
Yes, so I think Pfizer is going now for what they call a BLA. And so I think they'll be the first to try to get a formal license, and then Moderna would follow. Novavax hopes to, which is a protein vaccine, hopefully will put their vaccine for emergency use authorization perhaps in July. So that could be the next vaccine that would be approved under EUA. But I think the first one will be Pfizer, the second one will be a Moderna.
And how does that change things?
Well, then, I think when you have a BLA, then, I forget the difference between the emergency use and the BLA. And once it's a BLA, it's in the control of your physician, or, they can write a prescription. You can kind of get it anywhere. I mean, it's like any drug or antibody that's approved. So I think it'll make it easier to get broadly once it's formally approved.
So, because when I was speaking to journalists in Europe, they were very surprised to hear they could go to a pharmacy and get a vaccine in the United States. They can't. They have to either get it at a government center,
Yeah,
or prescriptions.
yeah. I mean, I think most of us that are in this really are most concerned about the rest of the world, and Europe and Africa and India and South America and Malaysia, and how we can continue to work. Scientifically, I think we've solved most of this. So this is really about manufacturing and delivery. And I think that once they have it, they have to probably deal with better delivery platforms, right? And make it easier to get. It's a shame, and we shouldn't be the ones that are fully vaccinated and other countries are way behind. I mean, I think we have a moral.
We're not fully vaccinated.
Well, no, but we're on our way. But I think all of us are most concerned about trying to get the rest of the world vaccinated with good vaccines. I mean, I think that kind of sits with most of us. That's why we're all focused on the variants.
So, okay, so we know that a lot of people have not been vaccinated. How do we get the rest of the people to get vaccinated?
That's a good question. I don't know what that number will be. Would it be 20 or 30%? I guess I would hope that maybe people would see that if you're vaccinated, you're free to, travel and go to bars and go to restaurants and go to movies and go kind of live your life freely, which is what we all used to do. And maybe without the fear of getting sick. And maybe that might show people that, that and be with family members and not worrying about passing it on to really young kids or really old people. And so perhaps that will convince people that getting the vaccine allows them freedom in their own life.
Right.
I mean, that would be, perhaps direct evidence to them that there's a great benefit to this, and you don't need to wear a mask. I don't think, other than that, I think you'd have to have people that can convey the message that people are comfortable with. And I think, right, that there's a lot of ways, shaped, and forms that could occur. It's not a one size fits all. It's not going to be people from the NIH and the CDC necessarily, but it might be just people in your community that you trust that are going to say to get it.
Well, the other thing that people said that if you can't fly without being vaccinated, if you can't go to a baseball game, you might decide, okay, I'll get vaccinated.
Yeah, I mean, I think that then you get into lifestyle. So I'm aware, I had read this, that if you go to a baseball game now, there's separate gates for people that are vaccinated.
Right.
And so they're going to see that the people that are vaccinated are moving through quickly and people aren't. So I think there might be a lot of tangible reasons why people's quality of life would improve with vaccination, rather than people like me telling them what to do, but letting people make their own choice.
All right, so let's get back to some questions. And at the end, I'm going to ask you, I always ask my guests, what do they do? Um, and, what is the probability that some of the breakthrough cases are actually cases of people who get dosed the vaccine that were not stored properly? Who knows?
That's an unknown. So the breakthrough cases, the question is how you define those. Are those breakthrough cases that are symptomatic? Or are those breakthrough cases where they're getting tested constantly and someone says you're positive, but you're asymptomatic? And so it's, quote, a breakthrough case, but the vaccine basically worked. The only issue, as I said at the beginning, is in my mind, is if you're PCR positive, you're feeling great, are you actually infectious? Those type things of not stored properly and things like that, I think are low risk. It probably happens. Could happen, but I imagine that's low risk.
So if someone wants to know, okay, we kind of asked this question when we know if a booster is necessary. So it said, should people who were vaccinated early in 2021 expect to still be protected by the November, December holiday season?
Yeah, so I spent a lot of time on that. I believe that you'll have immunity that potentially would protect you against severe disease and pneumonia and lower airway disease. The question is over time, might you be susceptible to kind of upper airway infection where you would convert and be PCR positive and be mildly symptomatic? So you don't go to the hospital, you don't get very sick, but you were able to contract the infection. So there's data from Pfizer that suggests that at 6 months, you still have very solid protection. But I think, at 12 months, we don't know yet. And it's conceivable toward the end of the year, there might be a recommendation about whether you would get boosted sometime in the fall or the winter that would carry you through. And as I said before, the data on boosting is quite extraordinary. And so,
but one thing I read is that don't ask your doctor for an antibody test if you have the vaccine, because it doesn't really measure the vaccines.
Well, you can get a test to measure antibody. The question is what does it mean?
Okay.
If he comes back and says you have a titer of 1 to 100 or 1 to 300 or 1 to 1,000, we don't have a metric that yet. You'll have one in a month that will say this amount of antibody looks to be protective. At that point, then it's possible that tests could be done to say, do you have that titer that would indicate that you're protected? And so it's possible that people with cancer or immunocompromised that are getting vaccinated, they might be monitored to see where they, have they responded to the vaccine and do they have what we would consider to be a protective titer. But I don't think it's likely or practical for the entire population to be getting their antibodies measured frequently. And right now, we don't know what those metrics would mean.
Okay. If there was a breakthrough infection in an elderly or pre-existing condition, fully vaccinated person, how effective is monoclonal antibody therapy in preventing hospitalization or death?
Yeah, so that's a good question. One thing I can tell you is if you are my age or, if you're on an age or have any pre-existing conditions and you're infected, I think the monoclonal therapy is something that you should seek. It's something that is very safe and it has a very high likelihood of working, especially early. So it's very high reward and there's really very little risk. The biggest issue is that it's underutilized. I mean, I think the companies have made it and provided it, but it's underutilized. But I can tell you that the primary basis of all vaccines are to induce antibodies. Some of the vaccines also induce T cells that could have a role. But if you think about it, if you're giving a large amount of antibody, it's going to be able to shut down the infection. So those of you who know people that might be suffering from a severe infection and they're elderly, the antibody treatments are really important. And that would be the first thing I would recommend.
So how many adverse reactions have been reported on the Moderna vaccine after second vaccination? And how does a doctor or patient report possible adverse reactions and to whom? Well, I can kind of answer that. I mean, I signed up and I got a weekly email from the CDC, a text message saying, how are you feeling today? And he's, so.
Yeah, so I don't know the exact numbers. In general, after the first immunization, I think having kind of local soreness at the site is quite common. After the second immunization is when most people feel more systemic, reactogenicity, where they can feel a bit sick, probably within a 12 to 24 hour period. And, it might depend if you're young or you're old, male, female. I think people react differently. I felt nothing. My wife felt nothing. My kids felt lousy. And so I would expect that after the second immunization, you might feel, some side effects. The other thing that if you've been infected, you should expect to feel more symptoms when you get your first immunization if you've been infected before. So that's the only time when the first immunization perhaps might be as bad, if not worse, than the second immunization if you've had prior exposure, because you've already had an immune response and now you're boosting it. So for those of you who have been infected, expect, at least with the RNA vaccines, one, a very good boost, but two, you might feel ill at that time.
So there are a lot of myths about the vaccine reactions. One person said females seem to get worse reactions. Another person said if you don't get a bad reaction, that means you don't have a very robust immune system.
Yeah. So I think we all worry, that if you don't have a bad reaction, you're not going to have a good response. But I think there is certainly data that females might have more reactogenicity than males. And so it might be idiosyncratic. If you haven't had a side effect to either vaccine, you still remember that both Moderna and Pfizer are close to 94% effective. So I think you have to be guided by the statistics and the data.
I did not get any severe reactions except pain in my arm. But Paul Offit told me that when you're older you don't get it, but then he got a terrible reaction after the second Pfizer vaccine.
Yeah.
What the heck?
Yeah, no, it's like, yeah. I mean, I was frankly concerned myself when I didn't feel anything after the second boost.
But you trust the science, right?
I always trust the data.
Okay. So one of my friends asked about the Yankees, but that was discussed very early on.
The Yankees?
Yeah, we discussed that already.
Well, that's what happens if you're testing people every day.
Right.
And they're feeling good, they might have contracted it. And the real issue is whether that is sufficient to spread it to somebody else. But obviously they were all asymptomatic.
And how do they come up with the numbering of the variants? Like you talk, B1, all these things.
I don't know that, how they all begin with a B. Yeah, I don't know that. The variant in UK is B117. The one in New York, I think, is 1526. Yeah, I don't know how the nomenclature is designed.
I remember someone said to me, I guess Moderna called it Moderna because, it's modern. I said, no, it's because it's mRNA. That's all I said. Okay, so you don't always know. We keep hearing about the percentage of the population who have been vaccinated, but not the group added to the people who have already had COVID, whether or not they were vaccinated. Isn't that an important metric when it comes to determining herd immunity?
Yes, absolutely.
Okay,
So I think you need to deal with who's been vaccinated, and then there might be, I would imagine, I don't know exactly, but I would imagine 20 to 40% of the country might have been infected. And so the question is, should you be adding the people who have been infected to the people that have been vaccinated into your calculation for herd immunity? So that's my area of expertise. I will tell you, though, that if you've been infected, depending on the severity of infection, you certainly have generated likely an immune response, and many of those people may in fact have resistance to a secondary infection. So in some way, shape, or form, they might contribute certainly to herd immunity if they've been infected.
Okay, so I live in New York City, as most people know, because I talk about it a lot. Everyone's wearing a mask where I am, and outside. I mean, you have to put on a mask if you go into a store. But should they be if they're fully vaccinated? Not according to CDC.
No, I mean, I stopped wearing a mask. I can tell you, I went to New York 2 weeks ago, and this is when I picked up the cold.
Okay.
So I didn't wear a mask. I went walking around Central Park both days. I think I had my first meal inside, and I wasn't wearing a mask outside. And ironically, I think this is where I picked up my cold. But no, I think that if you're vaccinated and you're outside, there really would be no reason to wear a mask. I guess the recommendations are is if perhaps you were at a concert and it was densely populated. Would you be wearing a mask? I mean, I think your risk of getting infection in this window of this time frame after both vaccinations is relatively low. And so I think certainly outside, that risk is astronomically low. Now, if going into a store, into a restaurant, I think when you sit down, you're certainly going to take the mask off and have dinner. So I'm myself, I'm comfortable being in a restaurant and without a mask on after having received the vaccine.
Indoors.
That might, yeah. I mean, I think that might, over the fall, there's a chance I'll get a boost to make sure that, I maintain a very high level of immunity. But yeah, right now, I think, especially for certainly outside, there'd be no reason, in my mind, to wear a mask.
Okay. What about indoor gyms? Because everyone says, the doctor says,
Yeah, I mean, indoor gyms is closer. I mean, again, if you've been vaccinated, you should have very low risk. I mean, if you feel more comfortable wearing a mask, but if you look at least with the Moderna and Pfizer data, the Pfizer data looks pretty good over several months. You're in a time frame that you still likely have protective immunity that would limit you from getting, any type of severe or respiratory illness. Maybe if you went to the gym every day and you got tested like the Yankees, you could convert and be PCR positive and wouldn't know it. But I believe that the data all suggests that you have a high level of protection.
okay. And did you fly to New York?
I drove actually, but I have no problem flying. I would have not a problem flying at all.
But you are required to wear a mask on the flight.
Oh, sure. No, I mean, I think if you're going into airports or you're going to trains and things like that, I think, you wear a mask, but underneath that mask, I think you have, high level of protection. But I think, yeah, I mean, I think you can maintain, we're probably gonna have to do that for a while. We'll see where the variants go. We'll see where herd immunity goes before that decision is made, where people would be able to take their masks off, in public places like that. I think a lot of the concern remains of whether over the next month, the hope was to really get a large segment of the population, or as many people vaccinated. And by doing that, you prevent the spread of variants. So I think all of us were worried is that before we tell everybody to take the mask off, that we, as many people vaccinated as we could, to limit the spread of virus and perhaps some of these variants.
So last week, as you know, last Thursday, CDC Director Walensky said, the science says, take off the masks. 2 days before, she was telling, got to wear a mask, got to be careful.
Yeah.
And these are confusing. These are confusing messages.
Yeah, I mean, listen, I think that the goal for the public health officials is to get as many people as vaccinated as quickly as possible, especially to protect them and protect the spread of variants. And so I think that they probably wanted to make sure before they made those formal recommendations that you had, as that there were vaccines available for people to get vaccinated. But I think as the data came in and they started to see that after 2 vaccinations, there was very little direct threat to people getting sick. And some of the Pfizer data suggesting that there was no asymptomatic infection. I think when they started to look at all the data together, I think it became clear that the recommendation made sense. I also think that recommendation is not only grounded in science, but I think it allows people to say that if they've been vaccinated, it might encourage people to get vaccinated based on the science and based on the ability to start to go back to the way things were.
So do you think we're, I mean, some states are lifting mask mandates totally. And do you think that, I mean, everything's opening up. Do you think that's a good idea?
Well, I think the risk is to people who haven't been infected or haven't been vaccinated.
Right.
So if you're a governor of a state where not a lot of people have been vaccinated, then in opening everything up, are those people at risk of getting ill?
Okay.
And I would worry about them getting sick. If you have a state where most of the people are vaccinated, then there's a high likelihood that they're not going to get infected and their hospitals aren't going to fill up. So I think it'll be interesting to see over the next several months what happens as things open up. Will there be pockets of areas where there might be increased infection in those areas where people aren't vaccinated? We'll have to see.
So the only vaccine approved for children 12 to 16 is Pfizer's vaccine.
Yeah.
Is Moderna going to get that approved?
Yeah, so Moderna's working on that now. And then Pfizer will start to, I think probably, hopefully in the next 6 months, work on an indication for younger kids. I think probably, on the age of, I don't know, 1 or 2 to 10. So I expect Moderna to follow Pfizer quickly. And then we start to move down in age.
Right.
So I think over the summer, what's good is that if you can get it to 12-year-olds, you would have middle school, high school, and colleges pretty well covered.
Okay. So I have very young grandchildren. They will not be getting vaccinated for a while, but I'm looking forward to seeing them. And
Yeah,
I guess,
exactly.
and I guess I can see them indoors because I'm vaccinated.
Mhm.
Um, what do you, so will the COVID-19 vaccine be a annual vaccine, you think?
It's possible. We'll see where it goes. I think I'm encouraged by the immune responses I've seen after the boost. And I'm hoping that maybe when you get the boost, if the immune responses stay high over a long period of time, and we don't get new variants that are resistant, then you might, have some greater durability where you might not, in fact, need it every year. We'll just have to see. But yeah, it'll be dependent on two things: the amount of antibody required to protect you, the development of new variants, and what's circulating. So I don't know. It's probably too early to make that proclamation. I would make. I do believe though that we should get it in the next 6 to 12 months. It's likely that we'll all get a boost, and then we'll see how long that lasts for. But
Do you think that will reach President Biden's goal of getting 70% of Americans having the first dose by July 1st?
I hope so.
Okay.
I think they've made vaccine, there should be enough vaccine available. I think the president has done a great job at making it, not only getting the companies to make it, but to find ways to distribute it. So I think that if people want the vaccines, it'll be available. I don't think it'll be up to, that 20 or 30% of people decide whether, what brings them to ultimately decide and get vaccines. I think some statistic like 80 or 85% of people over 65 have been vaccinated. So you've
Yes.
gotten the high risk population. So you're now starting to move down. But I think they've done a fantastic job in terms of distribution.
I have a friend in France who got the Russian vaccine because that's all he could get. What do you think of the Russian and Chinese vaccines?
Yeah, so early on, I actually was very intrigued, and I told people I thought what the Russians did was very clever. So what they did is it's basically the Johnson and Johnson vaccine.
Mhm.
We don't know exactly, is it the insert? We think we know that it's which spike insert, but it's a Johnson and Johnson, and then they take another adenovirus, a different one, and boost people with that. So it's what we call a heterologous prime boost. So I thought it was actually clever. And I wasn't necessarily surprised that it was working. And so, I think that the immune responses there might be better than the immune responses from the inactivated vaccines from China. As you know, there's two vaccines from China. There's Sinovac and Sinopharm. Sinopharm, I believe, is the one that's been used in the Middle East, which had been reported to have about 70% efficacy. I think Sinovac is in South America. They've been reported to maybe have 50% efficacy or less. I believe that those are the least, in terms of immune responses, those are probably lower than all the other vaccines we've talked about. But they've made them available and distribute them around the world. I mean, I think, frankly, if we could break the cold chain and you could ship RNA vaccines, refrigerators and things like that, that would be a better solution. I think there's no doubt that they're more immunogenic and they're more protective. So if you ranked them, it'd be Pfizer and Moderna. I think that the Novavax vaccine is probably very good. Then you kind of have Johnson and Johnson and AstraZeneca, and then I think the vaccines, the inactivated vaccines, probably fall a bit below that.
So Boris Johnson is interesting, case history of a person who was the kind of denier of COVID, then he got COVID, and then he had a goal of getting everyone vaccinated with the first dose and not worrying about the second dose. What do you think of that approach?
Yeah, well, I think, they I think it's an interesting approach. I think they were able to, by doing that, you got some immediate immunity. I think the immunity is not as good as after having a second shot, but that, a lot of people have argued that it makes more sense to get as many people as possible with that first dose and then bring them back for the second dose when it's available. I think that with the variants, it's really important to make sure they get that second dose. But COVID, having spent my life doing vaccines for malaria and tuberculosis and HIV, which are very, very difficult vaccines, COVID is a much easier vaccine, judging by the fact that almost everything has some efficacy. Even in an inactivated vaccine from China, you have the Russian, you have all these other vaccines that are inducing a little bit of antibody, and yet you can get, by and large, reasonable protection against severe disease and death with mostly every vaccine. So you don't need, such robust immune responses to have a biological effect. So I think in that vein, having everybody get a vaccine into them made sense to them and seems to have worked. But I think it'll be important that at some point they do get their boost, especially if the Indian variant or the South African variant were to start to spread, where I think the single shot would probably be somewhat less effective. That's been shown with the Pfizer vaccine, that the single shot is much less effective than 2 shots against the South African variant.
Okay. So I want to talk about your work with malaria.
Yeah.
Because mosquitoes kill more people than any animal in the world. I have 2 friends who had dengue fever and were very sick.
Yeah.
And so are we going to have a malaria vaccine?
Yeah, well, so I had spent a long time working on the malaria vaccine, and I made a strategic decision. I'm still working on the vaccine, but what we made is we made a monoclonal antibody to be used to prevent malaria. So the beauty of vaccines is when you give a vaccine, your body makes antibodies, and that over your lifetime, your body will continue to make antibodies. So you don't need to keep getting boosting, right? So malaria, turns out that the current vaccine, the best vaccine that's been studied, you need 3 shots when you're 5 to 12 months of age, and you get about 50% protection at 1 year. And then if you get a boost when you're 18 months, after 4 years, you're about 36% protection. So that's the data. And so that's not that efficacy is less than most of our other vaccines. And so I made a monoclonal antibody. And so the disadvantage of a monoclonal antibody is if I gave it to you if you were traveling, instead of you taking pills every day, if I gave you just a shot of an antibody and that lasted for a month or 2 and you were traveling with your family, you don't want to take the pills every day, that's a pretty good solution. Or what we're testing now in Mali is it rains for 6 months. So it starts in July and it ends in November. So suppose the beginning of the rainy season, you gave someone a shot of an antibody, and over that 6 months, it provided you complete protection. Now, what happens when you give someone an antibody is it eventually washes away, because unlike a vaccine that has trained your own B cells to continue making antibodies, the antibody itself is just a protein and goes away. So it's not meant as a lifetime intervention, but it could be an intervention for 6 months. So you can imagine if you could stop kids from getting malaria during that 6 months transmission with just a simple antibody, that might be very useful. And then finally, if you wanted to eliminate malaria, you need to have something that's very highly effective with only 1 shot. And again, we think an antibody might be a solution. So before the call, I was on the call with Gates discussing this. There'll be a meeting next month where antibodies will be highlighted. And today I just received notification an hour ago that our paper on the antibody was accepted into the New England Journal of Medicine. So this is a potential new intervention to prevent malaria. It's not a vaccine. And so we'll see. Malaria is a really difficult problem. The most common cause of death in infants and young children in the world in Africa. It's about 200 to 400,000 deaths. 300 to 500 million people get infected. So I got diverted to COVID over the last 15 months, but at the same time, I still work on vaccines for TB and try to make interventions for malaria. So we're excited to kind of get back going and try to see if we can move the needle for malaria.
Okay, so I want to thank you.
All right, sure.
Next week, my guest is going to be Andy Norman, who wrote a book about mental immunity and kind of boosting your mental immune system, which is interesting. And that'll be on May 26th.
Okay, thank you.
Awesome.
Take care.
Someone wrote in, I think Dr. Seder needs some chicken soup. So
I think that they're 100% correct. All right.
so thank you for taking the time to talk.
They don't have a Zabars in DC yet. All right.
We'll have to get one.
Okay.
Take care, and thank you, and everyone be safe, and
all right.
please get vaccinated.
Okay. Yes.
Good night.